and all the dignitaries who are present here to attend our third session on fermentation technology. It is my immense pleasure to welcome all of you. Today's speaker, Dr. Veena Gayatri, Assistant Professor, Department of Biotechnology, Stella Maris, is here with us to share her expertise about introduction of media formulation for lab and pilot scale, parameters responsible for optimization, and a detailed description of the different components of the medium that has to be added to the fermentation broth, further to conclude a systematic approach for preparation of the fermentation medium. It is my immense privilege to introduce this immense speaker who is my close friend to you all. She has 7 years of research experience and 10 years of teaching experience to her credit. Her area of interest are concepts related to environmental biotechnology, wastewater treatment, integrated method for recalcitrant removal. She has received a patent for a method for biodegradation of chlorophenols by halo-tolerant bacteria consortium during the year 2017. The projects completed by her are Tamil Nadu State Council of Science and Technology Student Scheme 2019-20 for biodegradation of organophosphorus pesticides, chloropyrophos using bacterial consortium, its metabolites in vermi remediation in contaminated soil 2020. Seed money received from the management of Stella Maris during 2018-19. Tamil Nadu State Council of Science and Technology Student Scheme 2018-2019. A comparative study on degradation of PHA by earthworm and its associated microbes Students Project Scheme 2019. DPT sponsored full scope maximum usage of Foldscope to apprise South and Northeast Indian farmers on the relative abundance of earthworm species associated microflora, for which she has received a grant of 8 lakhs. UGC Minor Research Project awarded in the year November 2016 for the concept reactive dyes of textile dye effluent degradation by aliphatic bacterial consortium, and for which she has received the financial assistance for 2 years of rupees 4,70,000. Her professional recognition, awards, prizes, credits, and fellowship received are UGC Fellowship for Meritorious Student in Science. Awarding agency is UGC Year 2005. Scientific Excellence Awards. Awarding agency Biozone Icon Award Year 2018. 11 Best Paper Awards in different national and international conferences from different colleges and universities throughout India during the year 2014, 16, 17 and 2020. To her credit, she's got 45 international journals in reputed journals with good impact factor, 4 book chapters, to mention few, Handbook of Research on Inventive Bioremediation Techniques, Chapter 18. She has also presented in 13 international and 35 national level conferences. She is currently guiding 3 PhD scholars and she has completed 50 MSc student projects. She has been very actively in organizing conferences and workshops, are about 6 national level conferences, seminars, workshops organized under DBT project scheme, workshops organized and conducted under DBT full scope scheme 2018-19. And she's also organized various workshops at state level and national level. She's an expert in external audit for the Academic Audit Department of Biotechnology, New College, DG Vaishnav College, to name few. Ma'am, we are immensely pleased to have you among us, ma'am. Now the platform is yours.
Thank you, Bharati ma'am. Very good morning to one and all present here. I would like to thank the principal, the vice principals of Shift 1 and Shift 2, and Dr. Bharati ma'am, head of the department, and the staff members of the department who has encouraged me to present on this webinar series on fermentation technology. I'll be with you for today and tomorrow's session. So there's going to be a lot of continuation of today's work for tomorrow as well. I believe you're all able to see my PPT, ma'am. Sir, it's visible, sir.
Yes, ma'am.
Yeah. So I'm going to talk first on media formulation from lab to industrial scale. First, I just want to tell you why we have to design a media. As we all require nutrients, as we all require energy, we need food. So, microbes also need their food and they need in the form of nutrients for their growth, for their metabolism, and their reproduction. If you can look into a media, for the people who are undergraduates, I think you all started with preparing a media. People are non-biology, and if you still you're attending it, I just wanted to say that it is actually a nutritional requirement. When I am talking about fermentation technology, we are talking about the concept of what fermentation is. Fermentation is actually a metabolic process where the microbes utilizes the sugar which is present there and they convert that into useful products and they release this in the absence of oxygen. So this is how the fermentation is defined. Basically, when you look at fermentation, it can, especially for industrial production, it is about bacteria, and I'm taking fungi, it can be yeast cells or the molds, which are actually involved in production. Okay, so what is the main composition of a media? I'll be talking into a detail about what we're going to go in and do it in a lab scale, and further how we are going to go into a higher scale up of the same media. Basic requirement is water, where we're going to dissolve any of the other ingredients into it, and then we're going to add a carbon source, a nitrogen source, and few minerals, vitamins, and oxygen. Oxygen, especially in case of aerobic bacteria. So it is very easy to design a media when you have simpler, pure compounds for a small scale. But imagine when you go in for a larger scale, it is little difficult because in a conical flask, we can control the condition and as well as the amount of media what we are going to use is already defined. But when I'm going to the large scale, it is very difficult to hold the same media what I have prepared in my flask. There are two major components which are present in a fermentation media. The first fermentation component, the major component is water. Whatever the source you take, the carbon source, nitrogen source, salts you take, or the buffer, this has to be dissolved in water. Next is the carbon and nitrogen sources, which are badly in need for the growth of a bacteria or a fungi. There are some minor components like inorganic salts, you have growth factors, vitamins, and minerals. Use anti-foaming agents which has to be added for an industrial production. This is not actually added inside a conical flask. And then you have a buffer and gases and enzymes. Some of the fermentation requires precursors. So I will be taking in detail about and at least give an outline about each of these media, how we are going to design and how are we going to go for a scale up. Okay, there are different types of media. See, when take about the fermentation itself, for a production media, we need to formulate the media. I will explain you what formulation is, but before that, there is two types of media. I think you can see that the one which is yellow in color, which is a fermentation media. This is actually large nutrients and industrial production, and it is economically feasible media which has to be designed. But when we use a media in the lab, it requires very less nutrients. And this is especially for the growth and as well as for the inoculum preparation. Other than the fermentation media, in addition of this media, we have divided them into two parts. Like no, any media if you take, it is defined media or undefined media. You can see the defined media is actually a synthetic media where we know the composition of it. But when you look at the crude media, we don't know the composition. Along with the defined components, we add extra components. Again, I'll be explaining you all this in detail. I'm just giving you an outline here. Okay, so what is the criteria for design of a media? First thing is The media, whatever you're going to prepare in the small scale, should support the large scale production. And imagine whatever we are going to do in the lab, it should have been a application in the lab. No, from whatever from the lab has to have the application in the field. That is going to be your pilot scale. So you should have a maximum accumulation of your product, which is going to be developed, and you should have very less amount of unwanted products which is going to be developed from that. But how do I identify this? I have to select the correct media. For example, I take antibiotic production, I take enzyme production, I take a primary metabolite, I take a secondary metabolite. For all these, I have to select the media in a proper way. There are five important things which a person has to think as a biotechnologist to look into how I can select the media. First thing is, I should have a reduced growth and highest recovery rate. Next is, I should have know the colony morphology every in between time intervals. Either it is going to be a small scale production or for the large scale production, whether my colonial morphology is going to be there or not. The third one is targeted organism. For example, I am using Saccharomyces cerevisiae. It should not inhibit the growth of my organism. The organism has to be there till the end of my fermentation, at least till the decline phase. Next is the failure to inhibit the competitive flora. I should not allow any other contaminant to act on my targeted organism. And please remember that the shelf life of your prepared medium is very, very important. Imagine you're going to prepare a nutrient broth today. You're going to prepare a nutrient agar today. Sterilize it today, autoclave it, keep it back. When I am going to keep it in the fridge and going to use it tomorrow, it should have the same shelf life from the time I prepared till the end it is going to be delivered. Before I start, I'll be explaining about this inoculum preparation again to you tomorrow, but I just want to give you an introduction about this inoculum preparation because I'm going to add this inoculum into my media. This is actually the left hand side. If you could just look at, that is actually the fermentar, how it actually looks like. And yesterday also, both the sirs they have explained about different parts of the fermentar, types of the fermentar. So I'm not going to go into the details, and that is going to be taken in the afternoon session today. And the first thing we should have to know is the first generation culture has to be taken. Let us assume you're having the culture, the targeted bacteria or fungi or your yeast cells, which are actually preserved on agar slants. That is all from the first generation culture. Second thing is you have to take that organism, which is a first generation culture, and then add it to a saline water or a nutrient medium. When it is going to be into a nutrient medium, glass beads are actually added to the agar and it is shaken well and then transferred to the round bottom flask to a flat bottom flask. Why flat bottom flask? Because that will avoid the contamination and complete suspension of your media and growth of your microorganism will be encouraged. The third step is the microorganism from the flat bottoms flask will be taken into a shaker incubator in a smaller shaker flask. Initially, when you start with 100 ml, then you go for 250 ml, and then 1000 ml and 2000 ml. So this is how you do a scale up in the laboratory itself. The last fourth step is check the culture. The microorganisms which are present in the shaker will be acting as a seed culture now, and you should see to it that from the small scale you're taking up the inoculum to the larger fermenter. So completely check the logarithmic phase of the microorganism which is actually prepared in your lab and take up that to the large scale. This is what is an inoculum preparation. I'm just showing an outline of the inoculum preparation here. You prepare it in a small flask, then you go for a small scale fermenter. Once you are completely understood the optimization conditions of how you have prepared the media and how your microorganism is reported and how much amount of product is formed, then you go for the pilot scale. You can just see the diagrammatic representation here. The culture is taken. I'm just explaining the same thing which I have put in words few slides back. You can see the slide culture is taken with the inoculation loop. I go for 5 days of rotatory shaker and then taking the microorganism again for 2 days inoculation for a shaker incubator. And please remember that I think you can see that I have shown all that as three flasks. You should have as triplicates. The seed culture also always has to be prepared in triplicates. And then you go for 15-liter stainless steel jar fermentation. Once you are very comfortable, you know the organism is growing well, aeration is there, agitation is there, and then you go for 60-liter fermentation. The seed medium, after it is completely stabilized, it is going in for the large scale up. So this is how a inoculum is prepared by addition into the media, and we perform the production. Okay, coming back to the media, I have told you about the inoculum, how you have to prepare it from a small scale, how we go for the large scale. Now, the media composition, if you look at, water is actually the main component. Other than that, carbon, nitrogen, phosphorus, and sulfur. Phosphorus sulfurs are actually in very less amount, while the other minor trace elements are available. You have vitamins like biotin, riboflavin, which are added to specific industrial production, and oxygen. Some of anaerobic forms require initial aeration. Example, in case of beer fermentation, initial stages of the fermentation start with the presence of oxygen, and then you create an anaerobic environment. You stop the oxygen levels there. Then you have the buffers. I think everyone knows we have to control the pH of a fermentation. Either it is going to be acidic or alkaline. You have to decide based on the fermentation, and if it is neutral, you have to control it with the help of acid or alkali. An addition of anti-foaming agents, as I initially told you, we have certain precursors, inducers, and inhibitors. So I will be taking each one at a time. Okay, why should we require a carbon source? Carbon source actually gives the growth and for the product development, and it provides you energy. Nitrogen source will help in protein synthesis. So without protein synthesis, microorganisms cannot survive. Next is minerals. This actually maintains the osmolality and the buffering capacity of the medium. In few places, we require vitamins. The main function of it is the cofactors as we add to the enzymes, and they synthesize the product. And oxygen levels has to be maintained, which is actually called as dissolved oxygen in case of fermentation. And you have a precursor which is added to some of the fermentation products. This will yield a particular product, and it is free from another particular product which is going to be developed. That's why we add the precursors. Next is, I want to tell you why are we looking at these nutritional requirements. There are three ways in which the microorganism requires the nutrients. First one is specific requirement, elemental requirement, and energy requirement. Actually, this is a simple equation if you see how a process happens in a fermentation tank. You have a carbon source, you have a nitrogen source, and I'm imagining it is an aerobic bacteria, and I'm applying certain specific nutrients for it. When I take that equation, on addition of all this, at the end of the fermentation process, I can see that the cell mass, the product is formed. Along with that, I will evolve the fermentation with carbon dioxide, water, and heat. So the main elemental formula which is no, applied in for a microbial cell is C4H7O2 and N. So carbon, hydrogen, oxygen, and nitrogen is going to be in a ratio. I will show you in further slides why we require all these. And remember, specific nutrient requirement, especially for oxotrophic bacteria. You can see that they have to go in for a complex medium. I told you there's a crude medium which has to be prepared, and that will help you to define a specific nutrient. Okay, other than these, we require certain environmental conditions. Say, for example, everyone knows that the temperature, pH, and water activity. First thing is the growth temperature, on which the cell yield is going to be maximum. And you have to see that the optimum temperature of the growth has to be reached. And all this has to be done in the lab scale itself. You have to check the optimum temperature and growth of your bacteria at particular temperature. Next is water activity. This actually plays a major role in the growth rate. It is actually determined as OTR, oxygen transfer rate, because water is also going to contribute to the oxygen levels other than your DO, that is your dissolved oxygen. And then you have to check the combination of temperature and pH on the growth of the bacteria or fungi we use. So it is going to be environmental conditions are going to be a major important criteria for addition of medium into your fermenter. Okay. These media can be as intracellular or extracellular effectors. Okay, what I mean by intracellular, like no, intracellular refers to that the organism itself includes certain genetic material and they control the genetic information. Extracellular represents the physical and chemical changes. I think I just told you about the physical changes now, the pH, temperature, which have water content, water activity, which has to be maintained. That is actually the system design and the culture system. And chemicals that nothing but which is your nutrients that is going to be present in your media. Okay, what is the selection criteria for getting the highest yield of my product? So there are a few important criteria which you have to think. I think I already told you this. We always have to think, we have to use less amount of raw materials, but yield a maximum amount of the product. Or you should always represent it as how much is my biomass per gram of my substrate. Substrate is nothing but the ingredients which I'm actually applying it as nutrients to the broth or my medium. And maximum concentration of the biomass or the product should be achieved at the end of the fermentation. See to it that the maximum rate of product formation will be achieved and there should be very minimum yield of your undesired product. You cannot stop the fermentation in between and you cannot say I have so much amount of undesired product formed. Nor during the fermentation process, I cannot get out of these undesired products. They are always there with the medium. And always imagine we are not working on non-biological systems. We are working with bacteria, fungi, or even algae or any other cell cultures, tissue cell culture or animal cell cultures. All these are biological systems. So only 70% of this is being understood. Why they produce the undesired product? You can even genetically modify the products, genetically modify the organisms, but undesired product formation cannot be avoided. So please plan the media in such a way that undesired product which is going to be formed is completely minimized. And you should always see to it that the minimum problem of media preparation. The sterilization is most important criteria of your media, aeration, agitation, and extra purification processes, which will be dealt to you tomorrow on downstream processing. How simple you can extract your compound which is added to the media. And media also has its effect on design of your fermenter. What type of fermenter has to be taken for what kind of product, and as well as it depends upon whether it is a batch culture or it is a continuous culture, whether it is a fed batch culture. So all these has to be experimented in a lab scale before we go in for the large scale production. Okay, I am coming back to the media on explaining you what a defined media is and what is undefined media. When you look at defined media, it is otherwise called as synthetic media. Here, it is very simple. I know all the chemical compounds which are present in that, and you can see to it that the physiological studies are being carried out. Say, for example, the biosynthetic pathway, the regulatory metabolism, all this is activated with the help of defined media. Example, you have glucose, ammonium containing salts, mineral salts, pure vitamins. I know this is the composition. I just buy it from the company and then I use it. But there's something called as undefined media or the complex media. In case of undefined media, you can see that addition of a peptide to a defined media makes it a complex media. It is as simple as that. But here, we don't know the requirement of the microorganism. It can be as a single culture or it can be as a consortium. Based on the requirement of the organism, I prepare a complex media. This is the undefined media. Next is, how am I going to design my or formulate my medium? Okay, there are a few things which you have to consider before you start designing a medium. Think about the function of these ingredients. They should produce the growth, the product, and you should know about the organism and the substrate it is going to be produced. And imagine it is all psychometric calculation. It is not just saying that I am going to just prepare a media today and tomorrow I am going to get the product. Everything is quantitative and I have to calculate with the component with the help of its concentration. Say, for example, If I am saying enzyme, I am going to say it as so much amount of specific activity the enzyme is produced, and so much nanogram of the organism is produced for biomass of my compound, and I have to optimize the conditions. Optimization is the crucial step, especially I told you about environmental factors like pH, temperature, combination of pH and temperature, their DO levels, all that has to be optimized. Only after checking that, modifying that, you can design your media. So please remember all these probability whichever I told you for formulation of a media. Okay. Other requirements include, I think I told you about carbon, nitrogen, phosphorus, and sulfur. Minor elements are also required in the form of vitamins, where you have biotin, riboflavin, and you need to know about the buffers, the acid alkali additions to it, and anti-foaming agents. In the later slides, I will tell you what is the most important part of anti-foaming agents and how it controls the fermentation process. Next is the media requirement. Say example, you take carbon, nitrogen or oxygen or minerals which are present. It gets conversion of the biomass into the metabolic product, and at the end of it, I generate carbon dioxide, water and heat. So it is very important to check the products which are formed and which is the output which is going to be generated after the end of the presentation. Like no presentation of my fermentation media, how I am going to evaluate all these at the end of my product formation. And you can see here the main elemental composition of a microbial cell is, if you take carbon, it is about 48% of the dry weight, while hydrogen, I have not mentioned at the beginning, but in a very less composition, which is coming either from your air or from the oxygen which you supply, which will generate the hydrogen. And some of the minerals can release the hydrogen into the environment of the fermentation media. It is only 7%, while in oxygen it is 32%, and you have nitrogen which is 14%. And you have to have a clear knowledge about the elemental composition, and there should be of the media which is refined completely before the industrial production starts. But when you look at these raw materials, it is very, as I told you, defined media in a lab, it's very easy. You have the nutrient source, you just add it, you take it from Merck, you take it from Sigma Aldrich, and you do the production. But imagine a large scale fermenter. We are not going to do an antibody production for 1 kilogram alone. It is going to be in gallons. So you have to be very careful in checking the raw materials which you're going to add. That is the cost involved, the transport and storage of it, availability of it. It should be available throughout the year. I can't select a raw material today and say after 6 months it is not available. Throughout the year it has to be available, and ease of handling it. In what type of form my raw materials are going to be, whether it is in the solid form or in the liquid form. These are the factors of choice which you take for the raw materials. Next is, after I start my fermentation, the media is going to be sterilized. This sterilization process itself will have a thermal damage. This actually contributes to the damage of a media, where it inhibits the product formation. You can see that operational characteristics or the formulation or mixing, this viscosity of the characteristics may influence the agitation, aeration, foaming, and recovery of your product. So always calculate the rate of formation and yield per gram of your substrate. And most important step is the downstream processing, where the purification levels are very important. You have to have a high purity product which you are going to deliver to the community, and the range of impurities which is going to be developed, which is going to be separated, and potential use of generation of undesired product. I think I have spoken a lot about it. Of all that, most importantly, whether it is going to have some health implications and safety precautions which has to be taken by choosing such raw material. Okay, I'm going to go a little in detail about each of these components. As I told you, the first component which is going to be present is water. Okay, why water? Major component is it is because it dissolves all the salts and it affects on contamination. So you have to be very careful. And in some cases, mineral content of the water is also checked before it is added to the medium. I think when we prepare a medium itself, after addition of all the ingredients, you check the pH. Imagine when you're going to do it in the gallons of product production, in a fermentation level in an industry, and it has to be done in a very, a very pure and quality way before it is uploaded. Other than this, into the media, it is also used for heating, the fermentation, cooling process, cleaning process, and rinsing process. All these process involves water. So pH may be adjusted to get different types. Say, for example, you're preparing a beer. Mineral content of the water, it is very important as in the presence of brewing industry. When you look at hard water, it contains calcium sulfate as salts, and this is actually used for English Burton bitter beer. The the color of the water and as well as the carbonate present will give the color to the beer. So that is how much crucial it is to select the quality of the water and when it is going to be supplied. Another thing is it is an energy source. Say, for example, in an industrial microorganisms, most of the microorganisms are chemotrophs. I think everyone knows what chemotrophs are. I'm explaining again here. Chemotrophs are organisms which use chemicals for their food. They cannot synthesize on their own and they require chemicals for their food. Common sources is your carbon source like carbohydrates, lipids, and proteins. So these actually contribute as a whole for the microorganisms to multiply. Some microorganisms just use hydrocarbons or even methanol as a carbon source. There are some factors which you have to take into consideration to select the carbon source. First thing is the rate at which the carbon source is going to be produced. Whether we are going to look at a primary metabolite or a secondary metabolite. How does it influence this? And how much is the concentration of sugar I am going to have and that is going to be metabolized and produce the secondary or secondary metabolite. And the main product of fermentation process will determine the carbon source. The purity of the carbon source is very important in case of substrate production. Say, for example, the metal ions must be removed in case of a citric acid production cycle, or else it will completely affect the process of citric acid production. Other than this, you can see that when you sterilize, I think everyone knows a person with learned with biochemistry or microbiology or with biotechnology. When you heat the sugars, they turn black. So when you use it in the lab, they are filter sterilized. So they react with the ammonium salts or amino acid presence and they form black colored compounds. This will actually inhibit the growth of microorganisms. So it is very, very crucial in adding sugars into the fermentation media. Say, for example, you take starch. When it is heated during the sterilization process, it becomes gelatinized. And this becomes as a viscous fluid or a liquid which is going to be there. And this cannot be used as such as a concentration. In that case, they are used as sterilized syrups. You can sterilize them by filter sterilizing them through membranes, and then you can use it. So next we are going to look at what are these carbohydrates and how are they going to contribute. Say, for example, first is molasses. It is actually a byproduct of cane or beet. I think I told you in the beginning itself, when you are looking at a carbon source in the level of your media in the lab, it can be just glucose, fructose, lactose, which is readily available because we are just going to add it in milligrams or maximum in grams. But this cannot be applied to the industry. So you have to check for cheaper sources through which it can give you a product. So in a sugar production, a dark color viscous syrup, this is how it looks like. I've given a diagram there. You can have a picture, the picture, we can have a look at it. These carbohydrates primarily are sucrose and they have 2% of nitrogenous substance which are present in them. Some they even have vitamins and minerals. So these are actually used for both low value or high value products. Say, for example, in production of single cell protein, ethanol production, organic acids, amino acids, or even microbial gums. Some of these are even used for antibiotics, vaccines, fine chemicals, and specialized enzymes which are produced. Next is molasses. Molasses is again a very good carbon source. Example in the form of malt extract. You can get these as aqueous extracts. These are actually from the barley syrup, and these is actually used for cultivation of filamentous fungi, yeast, and actinomycetes. It has approximately around 90% of carbohydrate, and some have vitamins and 5% of nitrogenous substances. Other than this, they also have protein, peptides, and amino acids. And you have to be very careful using molasses during sterilization because it will start getting heated up very fast when you sterilize it. So they are always sterilized at very low temperature where there is no color change, and there should not be any loss of the fermented product at the end of the production. And you should see that these molasses should not produce any undesired or toxic product at the end of fermentation. Next is starch or dextrin. See, this is actually, starch or dextrin are used and metabolized for metabolism of amylase producing microorganisms, example, filamentous fungi. I think everyone knows we have maize as the main source for it, and you have tapioca, which is also one of the source of giving the, by the powdered form, or you get it in the form of syrups. And this starch is getting converted in the form of sugar syrups. As it is very difficult to sterilize, we add to the fermentation media in the form of sugar syrups itself. The next one is lactose. This is especially used in case of continuous fermentation, where you want to have a crude lactose which is going to be prepared, and you have sulfite based liquor. This is actually a waste product generated from paper and pulping industry, and this is used for cultivation of yeast products because you want to have baker's yeast which has to be produced in a very large scale. It is actually the debbings which you get back or the waste which is generated from the paper and pulping industry. The waste liquor which will have from these coniferous trees, they contain around 2 to 3% of sugar and 80% of hexose and 20% of pentose. So this sulfite based liquor acts as a very good source because they are very low in their pH, and you can easily adjust the pH with carbonates, calcium carbonates or calcium hydroxide, and this can be supplemented with sources of nitrogen or phosphorus. So we have other cellulosic material as well. I don't want to keep on be repeating certain important things. This is cellulose is mainly used for mushroom cultivation. This is available in larger quantities as lignocellulose, where few microorganisms use this cellulose directly, especially fungi. And then you use whey. Whey is nothing but the one which we prepare at home. You could have seen there will be a small liquid layer, a yellow colored layer, which will come at the top of your curd. In the form of in the cheese industry, this is actually one of the byproducts which is going to be produced after cheese is produced, and this is employed in dairy industry. It is very expensive to store and transport. But some of the products which are food related products today, as no, you have these yogurt, whatever we have it in our field for having the probiotics. This acts as a main source. They act as a main source. Other than this, you have hydrocarbons, which actually helps in organic acids, amino acids, vitamins, and so on. And you have alkanes and alcohols. Some of the microorganisms they require methane as the source. So you get that from alkane or alcohol as the carbon. I'm not going to go into detail on all this. I'm just listing out here. You have methanol, ethanol, fats and oils. All these also serves as a best source of carbon. And other than this, they use vegetable oils, maize, cottonseed oil, linseed oil, soybean oil, and it is always taken into account that very occasionally fish oils are also used for production. It is seen that a typical oil contains 2.4 times that of the energy and that of a glucose or your body weight. So one thing is you have to know that so much amount of oil is added for so much amount of production. So it is used only for specific production. These oils are used as it is very costly. All these vegetable oils and maize. And why do we use this? It is especially for fed batch operation where you want to have a continuous fermentation process in between where it has to be organism has to develop. And some cases they are used for antibiotic production where they want to make the organism grow well and produce highest amount of antibiotics. Next is the nitrogen source. In nitrogen source, you have corn steep liquor. This is a byproduct of starch extracted from maize. Example, this is actually the main raw material used in penicillin production. And you use ammonium salts. This is actually for the baker industry. Today, from the starts with NaCl, ammonium salt as NaCl, ammonium sulfate, which is starting from the baking industry for a cake, for a bread, for muffin, any type of baking industry, they use ammonium salts. You have urea, peptone, yeast extract, soybean, and nowadays they also started using pea oil cakes. All these serve as nitrogen sources. So I'm just showing all the sources of nitrogen which is actually being available where they are used in different ways as a nitrogen source to be part. Okay, what does nitrogen source do to the media? Say, for example, in case of inorganic nitrogen, they are supplied in the form of ammonium gas or as ammonium salts. Ammonia is actually used for control of the pH and as well as a major source. And it is actually used for commercial production of human serum albumin. Especially Saccharomyces cerevisiae is used for human serum albumin, and ammonium salts like ammonium sulfate, they are used for acid conditions as ammonium ions. These are utilized as free radicals and they are liberated. You have other nitrate salts like ammonium nitrate, ammonium ions, organic nitrogen, and amino acids. Each of these will have its own preparation and own shift in nitrates for an alkaline grip. So the metabolism is going to be carried on with the microorganisms. As I told you, without amino acids, their metabolism or their replication and reproduction cannot continue. So they act as a major source for their DNA synthesis. Okay. Now, what are these proteinaceous substances? How can we get these substances as a large scale production? When I'm going for a large scale production, as I told you, it can be a Stonsky flicker, soy meal, peanut meal, cottonseed oil, and distillery waste can be used, and sometimes yeast extracts. These are used as nitrogen sources. But using nitrogen source is highly crucial because nitrogen source has its own individual components. It influences the metabolic regulation of the microorganism. So always remember the assimilation of one component has to be checked every time before the next component of nitrogen is going to be added. So if you're going to add two or three nitrogen sources to the same media, each one should not inhibit the growth of the other or allow not do not inhibit the role of other nitrogen source. So always see to it that a single nitrogen source will help the growth of your organism in the fermenter. So as I explained you before, I am just listing out here the Kaunski liquor, which is very high quality of nitrogen, and it actually a byproduct from starch extraction. When we do with the maize, through that itself we can get the nitrogen source, and that can be utilized. And you have yeast extract, which can be in two forms: yeast extract 1 and 2. Say, for example, when you use yeast extract as a first form in a fermentation media, it is actually salt free and it is avoided for corrosions. This is especially used for bakers and brewing industries. In case of yeast extract too, they are available as liquid content, a paste or a powder. This is very rich in amino acids, peptides, and they have water soluble vitamins. Some even have glucose, which are derived from the yeast source. So when you are buying even for a lab scale production, check whether it belongs to yeast extract one or two types. High media supplies both the things. Even for the lab scale production, before we start the production, we can check in which media it is able to do better. And if it is going to be very corrosive, your production of going to be very corrosive product which is going to be produced, and it is going to affect your stainless steel fermenter. Please see to it that you go in for yeast extract one. Next, we have peptones. Peptones actually act as a major source, but one thing is it's very expensive. You can use peptones in case of small scale production, but when you go for a large scale, they are prepared by acid or enzyme and hydrolysis. Say sometimes they take it from meat sources, gelatin, some from casein, peanuts, soybean oil, and cottonseed oil. All these are used as a source, either as peptone or soybean oil. You can see that 50% of that will contain as protein, and 7% are non-protein nitrogenous material, and 30% are carbohydrates, and they have only 1% oil. So you can see each of the carbon source and nitrogen source, they have different ways in which you express the proteins. Okay. Now I spoke to you on water, main component, the major component, and then I spoke to you on carbon sources, how it can be done at lab scale and in the fermenter scale. Next, nitrogen sources. Next is about the anti-foaming agent. This is most important criteria when you look at anti-foaming agent added to a fermenter. I have given a small picture here. You can just see what a foam looks like. It is like a broth which is completely no, frothified. Okay, what happens when a foam is developed? I just want to explain that for a minute and then go on for the anti-foaming agents. These foams, when they are formed, they are formed by the microorganisms, probably during the product production or during the process when the nutrients are getting depleted or when they're utilizing the microorganisms are utilizing the no carbon and nitrogen sources and releasing this as bubbles. So what happens is they form a bubble and each of this bubble combine together and they form a bigger bubble. So they will have small ones accumulated together and that will lead to the foam and that will lead to the frothing of the media. And you can see that the schematic diagram which I have shown you into towards your left hand side, you can see that this will accumulate all the oxygen content present in your media. And if you're not adding any anti-foaming agents, the microorganism will suffocate, especially in an aerobic metabolism, they will suffocate and they will die because these foams which are formed will not allow the microorganisms to grow. Tomorrow, I will be telling you about how a mass transfer occurs inside an air bubble and the microorganism. That will give you an idea about why a single small bubble will inhibit the production of any targeted metabolite which you want to produce. So you have to reduce the size of the foam which is going to be formed. We cannot completely get devoid of these foaming agents which are going to be there. So you have to get devoid of it and you have to reduce the size of the foam. Only then your fermentation will get completed. So as anti-foaming agents, they add surface active agents. What they do is they reduce the surface tension in the foam and they destabilize the protein films. These foams are formed only because of the presence of proteins which are expressed by the microorganism itself. So what do they do actually? Hydrophobic bridges between the two surfaces will be broken. There will be displacement of the absorbed protein and rapid spreading of the surface of the film will be avoided. So foaming component in the media or some factor which is produced by the microorganism has to be avoided with the addition of these anti-foaming agents. The most common cause, as I told you, it is the media's protein itself or the protein which is going to be denatured at the air and the broth interface. So they form a skin-like structure that doesn't rupture readily. So you have to add the anti-foaming agents to reduce this foam which is being developed. Okay. Sometimes these foaming can cause removal of cells from the media, which lead to autolysis. The organism will lead to death and further release the proteins from the microbial cells. Imagine in between your fermentation, usually when you're adding the microorganism, you will be adding the microorganism in their logarithmic phase. The production will occur either in the end of logarithmic phase or during the stationary phase. After end of the stationary phase, there will not be any growth. So when the foam is actually developed in the middle of the stationary phase, the product formation will be completely affected. Further, the release of the protein from the microbial cell will increase the stability of the foam. So at an uncontrolled condition, there are both physical and biological changes which will start causing problem to your fermentation process. Let us take the physical problems alone. Say for I told you the bubble size will change. It will be small in between. Then it will join with the next bubble. It will become bigger, and again it will become bigger. So during the size change in the foam, there will be very less mass and heat transfer rates which are going to be there. And this will invalidate your process data because we would have calculated some psychometric calculation to develop so much amount of media is going to be added, so much amount of inoculum is going to be added. At the end of it, I will have so much amount of product. But if the product is not formed the way you have planned it, then this cannot prolong. Other than the physical process, imagine the biological process. First thing, as I told you about the autolysis, the organism will start dying. Second thing is the microorganism will start depositing on the upper part of your fermenter. So this will affect the probes of your fermenter, and there will be a lot of problems during your sterilization operation. And the air filter will become wet, and that is dangerous again for your another microbial infection. The targeted organism will die, and it will lead to the loss of the product. So you have to know about the patterns of foam which are going to be formed in the media, and then their microbial activity. Hence, anti-foaming agents make a very important part during your process. So, way to take up the foaming. Okay, I know these are going to be the process, but how am I going to handle it? Before I select the anti-foaming agent, I should have all this in my mind. It should disperse readily, and it should have a very fast action. And it should be active at very low concentration. I can't keep on be adding an antifoaming agent during the fermentation. Once I have added, it should be very rapid, and it should have a long time action. I can't have new antifoaming agents every time added to the fermentation broth. Next is, I should not have any non-toxic. It should be non-toxic to the humans or animals. Whatever the anti-foaming agents I'm going to have, it should be very cheap, and should be heat sterilizable. And I'm adding to the fermenter after the heat is being generated. It should not affect my media and as well as the anti-foaming agent. And there should not be any exchange of oxygen transfer. It should not hinder the oxygen levels present in my fermentation. Next is the anti-foaming agents which are commonly used. I have given here towards the left-hand side. You can just have a look at it. Those are nothing but small air bubbles. So first, initially that you see at the primary level, you can see that primary gas bubbles are white in color, and then the primary air bubbles give rise to secondary air bubbles. They become bigger, and now I'm adding the anti-foaming agent. So when I'm adding the anti-foaming agent, immediately they get cleared, and I'm able to see the liquid phase. I have given at the bottom, you can see after the addition of the anti-foaming agent, how there is a dispersion of my foam which is going to be formed. So I have given the list of anti-foaming agents. There are both synthetic and as well as natural anti-foaming agents are present. Actually, on the fermentation field, they are still using only synthetic anti-foaming agents, especially alcohols, silicones, polypropylene glycol, fatty acids, ester. All these are used as synthetic molecules. But when you look at natural anti-foaming agents like plant oil or with the mineral oil or with fish oil, they are used in very less amounts, but still they do have effect as anti-foaming agents. Next is I'm actually coming to the end of my session on precursors. So when some chemicals are added to certain fermentation, they are directly incorporated into the desired product. Say for example, I add constip liquor as my nitrogen source in the production of penicillin. It increases the yield. But when I'm adding a precursor called as phenethylamine, this is incorporated to have a side chain of my penicillin molecule. In case of penicillin, I have different kinds. I have penicillin G, penicillin H, penicillin C. All this I can't do it at one time. But addition of a precursor material, especially as I told you, phenylethylamine, will produce only penicillin G for me. Second thing, real acetic acid. This is actually one of the another precursor which can be added for penicillin production. This will eat another type of penicillin. So there is something called as traded penicillin and types of penicillin which can be produced by addition of these precursors. The next one is inducers. These are added in very few fermentation process. You can see that in inducers you have there are induced enzymes, and they are actually when you add these inducers, they are actually helpful for producing certain amount of product. Sometimes you need a mini only then the raw material get converted into the targeted material by the microorganisms. So substrates, if you see, starch or dextrin for amylase is added, pectin for pectinase is added, maltose is added for quinase enzyme. So inducers are very potent. Say for example, you have isovalonitrile that induces nitrolase. So you have these enzyme inducers which will help So the intermediates to not to be developed much, but their analogs or substrates can be developed. These are still used in the process on how we can manipulate the microorganism, not genetically modified, but still we can manipulate the microorganism and produce a targeted product of our interest. It is just to use the microorganism to take up the metabolite or your raw material and produce the targeted compound. Okay, I will be talking about how we genetically modify the organism and involve them in fermentation in tomorrow's session as well. I'm just giving you an introduction here on that. When I'm talking about GMOs, there are so many GMOs in the worldwide use today in an industrial production, especially for production of heterologous proteins of viral, human, animal, plant, and microbial origin. So in this case, you can see that all the organisms, especially yeast, fungi, which is molds, and then the bacteria, they play a crucial role of protein synthesis, and when they are added as a host culture. So there are certain amount of biomass which has to be developed and they have to be induced. Only for these conditions we go for genetically modified organisms. And the last part is about the inhibitors. See, sometimes I'm not into the direct metabolism. I want to redirect the metabolism for my microorganism. They don't have to take up the targeted pathway of their own. So in that case, I allow the inhibitors to act on it to target the compound, and they form the metabolite intermediates, and they allow the production of stop the production of undesirable product. Say for example, we have chlorination inhibitors like bromide, which minimizes the chlorotetracycline production during the tetracycline fermentation. So these are small techniques which are being induced in a fermentation process to check out whether the microorganism is able to make smaller products which are actually the regular metabolism is not followed by them. And next, they also play a crucial role in cell permeability. It affects the cell wall of the cell wall structure and increases the permeability of the release of the metabolite. See, importantly, when you look at some intracellular products produced from the microorganisms, say for example, you want to extract a surfactant, you want to take up an antibiotic like penicillin. So in that case, they are added with amino acids. So these are small inhibitors which will allow the microorganism to go in a different metabolism of our target. As first and law release the buffers. As I told you from the beginning, pH control plays a major role and buffer is always with the water. So control of the pH is extremely important for optimal productivity. Sometimes we even work with acidic pH, sometimes with alkaline. But most of the production today in the field, if you see, they are all linked with neutral pH. So addition of ammonia, you can add simple NaCl, NaOH, sulfuric acid. All these are added to control the pH of the fermentation product. So a compound may be added to the medium to serve as a buffer and may also serve can be served as a nutrient. So, for example, if I want to make the buffer to have a pH of 7, calcium carbonate will be added, and this will decompose if the pH is decreases. And there are some phosphates which are added as a part of the medium, and they play a major role in the buffering. And we have a balanced use of carbon, nitrogen, and they form a pH control in their buffering. And you can see that this can be done by addition of proteins, peptides, or amino acids present in the medium. Lastly, the chelators. We add EDTA, citric acid, polyphosphates. These are nothing but insoluble metal ions. So when you are, for example, when I am preparing the medium, I think when we have these ion compounds, metal ion compounds like iron, calcium, zinc, or manganese, it will precipitate in the medium. To stop the precipitation, we add these chelators. So these form complexes with the metal ions in the medium, which may be gradually used up by the microorganism. In that case, all the raw materials will be utilized by the microorganism. Other than that, there are few growth factors which are added to the medium, and you can see that these growth factors help the complete cell components to be replenished by the microorganisms, and they will be able to grow in a better way. So you can see that growth factors commonly used are vitamins, and they are specifically used for enhancing amino acids, fatty acids, or sterols. So if there are some vitamin deficiency, it can be often eliminated by careful blending of these materials. Sometimes cheaper multivitamins are also added in a larger quantity. So like biotin, thiamine, or calcium phthalate, all these will help in increasing the growth of the bacteria or the fungi which is being added, and which are especially or no, lacking certain criteria for vitamin production. So minerals are already there in your medium as a defined media or as a undefined media. But other than this, magnesium, phosphate, calcium are added in the form of cobalt, copper, zinc, so that that will enhance the organism's osmolality, and they will be able to survive in a higher way. So this is actually phosphate is added in certain kind of media in a laboratory flask. It is not higher than that in the mineral components. This phosphate is used as a buffering agent to minimize the pH changes found with the media. So I am giving a schematic diagram. You can just have a look at it. It is from the Singh et al. 2017. When I am developing a media, I have to follow certain protocols. Please start with a database of a carbon or a nitrogen as a literature search. Say, for example, I am going for an enzyme study. I have to know what are all the carbon and nitrogen sources they have given in the literature. And after, based on that literature, I start my work. I know my microorganism is producing that, I optimize such conditions. And then I carry out the experiment. Once I am significantly knowing about the carbon and nitrogen, then I go for the scale up. When I am knowing the significant component which are present there, then I go for a shortlisting of my optimization studies. Optimization studies plays a very crucial role, especially your environmental factors, the minerals present, the salt conditions which are going to be there, pH, temperature, pO, all that has to be accumulated. Once I know, I start adding it to the new medium. After I know so much amount of growth has been established, I go for the fermentation broth. Once I've gone to the fermentation broth, I again add the new medium. So based on whether it is a batch culture or a continuous culture, I design my formulator media and go for the higher scale up. And increasing the productivity reduces the overall cost of the product. So always check for cheaper sources, and a good stain is needed. I'll be talking on stain improvement also tomorrow. How to improve the stain before it is added to the fermentation media. So there's another thing. What he has quoted is one factor at a time. It is called as OFAT. I'll tell you what does it mean. Always check for one variable at a time. If I'm taking a media, I am checking for pH. Optimize the pH alone. Then I am taking temperature, optimize the temperature alone. When I'm checking with the combination, then have the optimize the condition of my pH and the temperature together. That will give you the effective OFAT. And nowadays, it's not just with what are the experiments what we are doing in the lab. There are so many softwares which are available. You can use a response surface methodology, artificial neural network (ANN), and genetic algorithm is there. You have Nelder meet Simplex. All these are nothing but softwares which are being developed today to optimize the conditions in the computer itself. You can, based on the microorganisms, you can use these softwares. Most of these are freely downloadable, and then based on the optimization, you can go in for the higher scale of study. So there is a gist of all the design and optimization which are there in the papers. Kindly read through the papers, have a targeted organism, and every targeted organism, I have given the reference here, you can just see that from 1992 till 2014, so many microorganisms and so many metabolites, each of which, what type of technique, what they have followed is also given. So before you go in for a production media or for a fermentation, design, formulate the media, and then go in for the production. Thank you for your patient listening. I think I have taken a little more. There's no other way. I have to explain everything. Thank you very much. I'm here to ask, listen to your questions.
Thank you, Dr. Veena Gayatri ma'am, for elaborating us about the various mediums involved in the laboratory scale product production, and how to scale it up to industrial level. Now I request Dr. Mahindra Kumar, Assistant Professor, Department of Biotechnology, to take over the discussion session.
Thank you very much, ma'am. I once
Thank
again
you, sir.
thank our speaker for giving a nice presentation and detailed informations about fermentation and media formulation. Ma'am, I just go through the questions and I'll post it here, ma'am. You can see an answer.
Yes, sir. Yes, sir. Okay. So there's one person asked about how is media addition controlled in large scale process. To Nilima Devi, I would like to tell you that in case nowadays we have all that which is automated controlled fermentation is available. But even if you don't have one, for example, if in our lab we have a 5 liter fermenter, how do you go about it is when I'm adding a media addition, I can check. There will be well, during the presentation itself, you would have seen there will be a pH probe which is going to be there, a temperature probe which is going to be there, and I will have a media probe which is showing the DO. So what will happen is when I'm adding the media, immediately after the addition of the media, the DO levels will be shown. But when I'm adding the inoculum to the media, there will be the dissolved oxygen because immediately after the addition, in few minutes of time, the microorganism will start multiplying. Okay, especially in case of aerobic bacteria, they require only minutes for them to multiply. Only thing is, and as I told you, we are not directly going into the lab scale from the large scale fermenter. We will be working on a small scale, checking the whole media addition, and then going back. It will take an acclimatization will be there as you are asking. There will be an acclimatization stage which will be there, and media addition when you're adding it, for example, using a 25-liter fermenter. 25-liter fermenter, we should have a head space, and you're going to add around 15 liters of your media. In that 15 liters of media, whatever you're going to add, you will be noting down the pH, temperature, DO, which is automatically recorded. Other than that, the components are already stabilized outside and then only it is added inside. But there are changes which can be there. There is a withdrawal loop which is available in between the fermentation process. You can withdraw the sample and check the growth of the organism and check the components which are produced from that. So growth OD, colony forming units per ml, all these are already available. Next question. Any permeability compounds such as penicillin or surfactants are mentioned in the PPT. Do they not affect the products that are released from the cell? Yes. Rajeshwari, one most important thing is, say for example, surfactant is not added for all the penicillin production. There are only at certain cases and very negligible amount of surfactants are added. And these surfactants and as well as deionized compounds, whatever we are going to add, they are actually going to enhance the production media and they will not affect the cells. And for this also, you have to do an experiment basis in the lab and check how it is going to be and then add it to the media. Next question. What could be the reason for non-growth of lactobacillus in fermenter but in the inoculum? Um, Adnan. I don't know how to spell your name. Sorry for that. But before you start the fermentation itself, you have to check the growth of the organism. The seed culture itself will show you how your organism is actually going to multiply. And after that, see to it that you are always having no only forming units per ml, which is put down in your MRS agar plate. Only when you get the viable cell count, not even the growth. Only when you get the viable cell count, then go in for higher production. If it is not growing in your small scale, never go in for higher scale production because the growth may not be enough. Probably, especially in case of lactobacillus, it's a slow growing organism. Hey, you're looking in for a bacillus in production. These slow growing organisms, a lot of growth factors. So optimize some of these growth factors and try. It will surely grow in your lab scale. Once the seed culture is developed, then go in for the higher scale up. Okay, I read the question. What all should be taken into consideration for preparing a media for continuous culture because we tend to add media continuously? Correct. First thing, Manigandan, you have to know whether your product can go for batch culture, continuous culture, or fed batch culture. And there are so many reports on the product which is going to serve you as the best. Once you get to know which is what it is going to be, okay, you have decided it is continuous culture. See to it that the same continuous culture you are able to work in the laboratory condition. You can even do this in the 1000 ml flask. Check continuous culture is when you are doing a continuous shaking, whether you are able to get the product. If it is not doing well in the lab condition, never take that for fermentation vessel because fermentation vessel is very crucial. You're using so much of raw material and so much of effort is needed. So always challenge everything at your seed culture level itself in your laboratory scale and then go in for fermentation. Will the expired media have a yes? Never use any expired media because it will surely affect the cell growth. Nutrients which are new itself can affect the growth of the bacteria. So never use a expired culture. How to find out which precursor or inducer is required for a fermentation media? Lokesh Sharma, this is a standardization protocol. First thing, what you have to do is you have to standardize your protocol in such a way that you have your microorganism grown with a precursor. And imagine when you are using a precursor, don't use higher quantity. Less quantity of precursor has to be used because these precursors and inducers itself can spoil the fermentation process and do not allow the microorganism. And how you standardize it is you have to do a standardization study. You have to do a standardization study. For example, let us assume you're using a precursor which is giving you food production in the lab scale. Sometimes in the large scale it may not give. Next question, sir.
Ma'am, that's all, ma'am.
That's all, sir. Okay.
There are no questions, but many have. I would like to just read few appreciations. One, Lokesh Sharma has
Okay.
said, very nice in-depth presentation with good examples. Lot many things to learn. Thank you, ma'am. And
I want to give more, sir. So much is there to talk, but the time is a constraint, sir.
yes, ma'am. One, Manigandan has told, thank you so much, ma'am, for a nice and detailed presentation. More than questions, we have a lot of appreciations, and they have
Oh,
thanked you for the clear
I think
presentation.
we're all happy with what I did, sir. I will not be boring because this is a lot into theory.
Yeah, yeah,
I
yeah.
think people will be appreciated better.
Yes, ma'am. All have appreciated you only, ma'am.
Yeah, thank you, sir.
Thank you very much, ma'am. Now
I,
I
I will actually tell one comment, sir. Whoever has come today, please kindly join me tomorrow also because tomorrow I'm going to talk about metabolite production. It is just a continuation of what I am doing today, and I'm going to tell you in the industry what type of metabolites are there and how it can be actually implemented as a production. So that will be and tell you about the mass transfer. So that will give you a clear outline about how from the lab scale it is directly taken into industrial scale and what are all the parameters which is going to actually affect the fermentation process.
thank you, ma'am. Now I request our head of the department, Dr. Bharati Ravikrishnan, ma'am, to say the vote of thanks.
Thank you, Veena Gayatri ma'am, for patiently
Thank you.
answering our questions, and thank you, Mahinder sir. It's a formal procedure for me to render my vote of thanks. It's my indeed a very proud privilege to offer an vote of thanks today. My heartfelt thanks to Honorable Speaker Dr. Veena Gayatri, Assistant Professor, Department of Biotechnology of Stella Maris College, who has been sharing her very depth insight about media formulation in both lab scale and industrial scale, and also very patiently answering all questions of our participants in spite of our few health issues also. And my heartfelt gratitude to our respected General Secretary and Correspondent, Mr. Manjit Singh Nair, who has been supporting in all our endeavors in his absentia today. Dr. N.G. Raghunathan, our principal, for his critical suggestions, vice principals of both Shift 1 and Shift 2, my dear faculty fraternity of Guru Nanak College students for extending their support in organizing and conducting this online webinar series. And finally, not but the last, to all our valued invited guests, ladies and gentlemen, my heartfelt thanks to all of you. I render my special thanks to my students, Mr. Manikandan, Mr. Balaji, Ms. Dakshita, Mr. Bonesh, Susan, and all my department colleagues, Dr. P. Mahalakshmi, Mr. Amal Ludraj, Dr. Mahindra Kumar, for support in bringing up this webinar series. Thank you all. Now, I request Dr. Mahindra Kumar to address the participants with few instructions.
Thank you very much, ma'am. Dear participants, our fourth session will be exactly will start at 3 o'clock today, and the links have been posted to you. Once again, we will post the links through the WhatsApp group, so that I request all of you to join for the fourth session and upcoming sessions also. The attendance feedback link have been posted in the chat box. Kindly submit your attendance feedback. In case you face any issues, you can very well, you're welcome to tell your issues in the WhatsApp group. I assure everything will be clarified and sorted out. Thank you very much. We'll conclude the session today, for now, and we'll meet you in the session 4, that is at 3 o'clock today. Thank you very much.